Okay, hi folks. So today I'm going to be talking about the specific immune response. So yesterday you were learning about the non-specific immune response, which was all to do with phagocytes and phagocytosis. And actually this response starts in exactly the same way. So there's the pathogen, and there's the pathogen's antigen, which is what makes this a specific response, because it's going to be making something to be specifically binding to that antigen. And here is a white blood cell. It's actually called a macrophage, which is just like a large version of a phagocyte that you learnt about yesterday. And it's going to be engulfing the pathogen in exactly the same way as you learned yesterday. But the difference is that when this cell has engulfed the pathogen, it takes the antigen from the pathogen and it puts it onto its own cell surface membrane. Look, it's presenting it. It's almost saying, look what I've got. Okay, so this stage where the macrophage is engulfing the pathogen and presenting the antigens is called antigen presentation. And this kind of kick-starts the recruitment of a load of other white blood cells. So some of the white blood cells that are involved are T lymphocytes. Here is one example of a T lymphocyte, and it has receptors on its surface, and the receptors on the surface of this T lymphocyte will allow it to bind to the presented antigen on the macrophage. Once it's done that, it's able to do something. Meanwhile, there's a load of another type of lymphocyte, the B lymphocytes. So basically, I like to imagine that they're all coming down with their slightly different shaped receptors. There we go. And they're all queuing up to present their receptors to find out which one is the complementary receptor to the presented antigen. Remember, the presented antigen is the one that was on the pathogen, which is like a little triangular shape in my picture. And the T cell here, the one that's bound, is able to produce a chemical. Okay, so these chemicals are able to select the B cell that has the complementary receptor to the antigen. So let's write that down. Chemicals secreted to select the B cell with complementary receptor to the antigen. And remember, it's the pathogen's antigen that we're really interested in, because we want to make something to be complementary to that. So in my little picture, I don't know if you spotted, but this is the one, this is the B cell that's got the receptor. This is the one that has to be selected. So the chemicals are going to be secreted onto this B cell. It's going to be selected, and then what happens is loads and loads of copies are made of it. Loads of clones, yeah? So it's just drawing some more of these B cells, and now the difference between this row of B cells and the row of B cells above is that all of these B cells have exactly the same receptor. They are all clones. So they have been cloned, they've divided by mitosis. Let's write that down. This stage is called clonal expansion. Expansion is to do with the increasing number of B cells that have all got the same receptor. And if we just go back, I've just put a little box around that, that's the name of the stage. And if we go back, I didn't mention what this stage was called, but this stage is called clonal selection. So let's just put that in. Put that in a little box as well so we can see. And we'll put the antigen presentation. So far we've got 3 stages, but we haven't yet got any antibodies. Okay, so it's the next stage that allows the antibodies to be produced. So some of these B cells here get made into cells that can make the antibodies. They're called plasma cells. And this stage is called differentiation because what's happening is the B cells are taking on a specialized role so that they can carry out a particular job, which is what cell differentiation means. They're specializing to allow them to be able to carry out a particular job. So some of the B cells differentiate into plasma cells, and it's the plasma cells that make the antibodies. So it's interesting to think about what kind of question you might be asked about this plasma cell and what kind of adaptations it has specifically. So for instance, if they're making antibodies, we know from your definition of an antibody that it is a protein which has a specific shape that is complementary to a particular antigen. So if it's a protein, these plasma cells will have to have lots of ribosomes, lots of Golgi body, lots of mitochondria, because it's an energy requiring process, making and secreting proteins. Okay, and then let's just have a look and see what these plasma cells do next. They make the antibody. Here are the antibodies being secreted. Obviously, they secrete more than one at a time. And these antibodies then enter the bloodstream and they go off to bind to any antigen that's complementary to it. And we'll think about what happens to those in a bit. Okay, so off they go. And if I just draw an arrow, we can see that they would go into the bloodstream and they would go all the way back up to any pathogen that hasn't yet already been engulfed and bind to it. And we'll think about what happens with that later. Okay. Meanwhile, you'll notice I didn't bracket off these B cells because these B cells are actually going to differentiate into something else. So some of them are differentiating into plasma cells, some of them are differentiating into memory cells. Okay, so let's just have a notice that the receptor that they carry is the same receptor as the B cells and the selected B cell, and it's complementary to the pathogen's antigen. Now these B cells, they don't do anything to begin with. So what they do is they stay in the bloodstream after the infection. So some B cells differentiate into B memory cells. And it's these which recognize the antigen if it's presented to the body again. Oops. Okay, so this is what underlies your immune memory. So let's just go through this again. If you get infected the first time, antigen presentation happens. Meanwhile, you know, this macrophage is only going to be able to engulf so many pathogens. There's still going to be hundreds of them milling around, hence the need for this army of cells and a load of antibodies to help round them up. So this happens, and then this happens. Antigen presentation, then the clonal selection, then the clonal expansion, and then the differentiation, and then the antibodies are made. Meanwhile, this could take about 5 days before you can make antibodies against the infection, during which time you're going to get quite ill. If you have the same pathogen enter your body again, instead of having to go through all of these stages, what happens is you just activate your B memory cells, and then they will differentiate straight away into the plasma cells, and the plasma cells will make the antibodies. So this is what underlies the immune memory, and it's the way that vaccines work. So vaccines take an inactive form of the pathogen, or simply just the antigen. You then go through all of these stages, make the B memory cells, and then if you are exposed to the infection, to the pathogen in future, you have the B memory cells and therefore the facility to make the plasma cells and the antibodies very, very rapidly. So if we just finish this little tutorial by having a look at a graph, okay? So this shows you the concentration of antibodies, your first exposure to the antigen, and we can see that there's a delay. This delay here is equivalent to 3 to 5 days, where you are making no antibodies because you're having to go through all of these stages the first time that you're exposed to the antigen. So eventually you start to make a few antibodies, but not very many, and quite slowly as well. Okay, but then what happens? The antibody concentration will go down, and if you get exposed a second time, what happens is you produce antibodies so much more quickly. Let's just go back. Because you have the B memory cells in place, you'll be able to make your plasma cells instantly and then make the antibodies immediately. And so what will happen is you'll make the antibodies very, very quickly, and you'll make loads of them.